Welcome to Pihu classes. Today we will learn cell membrane.
The cell membrane is an important structural element of the building block of life, the cell. Its main role is to define what's inside, the intracellular space, and what's outside, the extracellular space. It also regulates what comes in or out of the cell, and that's called selective permeability. The cell membrane is basically made up of a bilayer of phospholipid molecules. Phospholipids are amphiphilic molecules, meaning both loving. Now, the phospholipid is made up of three things: their head, which is made out of negatively charged phosphate, a tail, made out of two fatty acids, and a skeleton made out of glycerol that brings everything together. Their head is hydrophilic, meaning that it likes water, but meanwhile their tail is lipophilic, meaning it loves fat. These lipophilic parts also exclude water, so they're not just lipophilic, they're also hydrophobic. In water, phospholipids form a bilayer where the hydrophobic tails are oriented inwards, where there are no water molecules, and the hydrophilic heads are oriented outwards in contact with water. So the plasma membrane forms a wall with water on both sides. The cell membrane is also semi-permeable. That means that the membrane allows some molecules to pass through, but not others, and it's mostly based on the molecule's size, polarity, and charge. There are roughly 5 categories. Small and non-polar molecules, like oxygen or carbon dioxide, will diffuse through the membrane quickly. Small polar molecules, like water, will be able to pass through, but it happens relatively slowly. This is because even though the middle of the phospholipid bilayer is hydrophobic, the occasional molecule of water can sort of slip through because it's such a small molecule. Now, large and non-polar molecules, such as retinol, also known as vitamin A1, can also cross the membrane thanks to them being non-polar. But once again, crossing is really slow because the molecule is so large. Now, as you might guess, large polar molecules like glucose are unlikely to pass the cell membrane on their own. Highly polar charged ions like sodium, potassium and chloride, or molecules that possess a charge, like amino acids, stand no chance of passing the cell membrane. In addition to phospholipid bilayers, membranes also contain cholesterol. Without cholesterol, at low temperatures, phospholipids pack tightly together and become less fluid, and that makes the membrane brittle. Without cholesterol at high temperatures, the phospholipids separate from one another, and that makes the membrane leaky and weak. So the role of cholesterol is twofold. At low temperatures, it squeezes in between the phospholipid molecules and keeps them from packing too tightly together to keep the membrane more fluid, and at high temperatures, cholesterol pulls phospholipid molecules together, decreasing the space between them. So good old cholesterol makes the cell membrane fluid and durable no matter what the weather. This is particularly important for cells like red blood cells that have cell membranes that see a lot of wear and tear over time and get exposed to different temperatures. In fact, that's why red blood cells have membranes with even higher levels of cholesterol than normal. Cell membranes have various membrane proteins embedded within them. There are integral proteins that span the cell membrane bilayer, peripheral proteins, which are found on the inner or the outer edge of the membrane, and lipid-bound proteins, which can be found hanging out in between the phospholipid layers. Some of these membrane proteins are transport proteins, and they're examples of integral proteins because they span across the membrane. Transport proteins help move molecules that can't freely diffuse across the membrane to get in and out of the cell. There are 2 types of transport proteins: channels and carriers. Channel proteins open to form a sort of tunnel through the membrane, through which water and ions can flow right through. An example would be an aquaporin channel, which opens and closes to let water in. In contrast, carriers are very specific. They have a special binding site that only allows a certain molecule to bind to it, and they also have gates at both ends, which open sequentially. One example of a carrier protein is the glucose transporter called GLUT4. By default, the transporter is open to the outside, waiting for a glucose molecule to float by. When a glucose molecule binds to a special site within the transporter, the outer gate closes and the inner gate opens. The glucose then unbinds and floats freely into the cell, while the transporter will reset and await the next glucose molecule. There's also a special family of transport proteins, which are essentially enzymes. These transport proteins use the energy from adenosine triphosphate, or ATP, to actively pump various ions in or out of the cell against their concentration gradients. That gives them their name, ATPases. In other words, these ATPases push ions from the side with a low concentration over to the side with a high concentration. A classic example is the sodium-potassium ATPase, which pumps 3 ions of sodium out of the cell in exchange for 2 ions of potassium into the cell, both going against their concentration gradients. Cell membranes also contain cell adhesion molecules, or CAMs, which are integral transmembrane proteins that anchor the cell in place by attaching it to other cells. These cell adhesion molecules come together and create junctions called adherence junctions, or tight junctions. An example of a cell adhesion molecule would be the cadherin, a membrane protein whose job is to hold tight to neighbouring cell cadherins at adherence junctions. Think of them as Velcro attaching the two cells together. The tight junction is, well, even tighter, and creates a fully waterproof seal through the creation of protein complexes, and it sort of resembles rivets joining metal plates together, ensuring waterproofing. Cell adhesion molecules and junctions are essential for the structural integrity of multicellular tissues. They're like the mortar and the cell are like the bricks, together forming a really strong brick wall. Next are the cell surface receptors, which receive an input from the outside and transmit it to the inside of the cell. The input is usually something that other cells produce, like hormones, cytokines, or growth factors. So the cell surface receptors are sort of like the cell's mailbox, receiving news from the world. If a hormone binds to a receptor, the receptor changes its shape, and that triggers a chain of events within the cell. For example, when there's a lot of glucose in the blood, the pancreas secretes insulin, which is a peptide hormone that binds to insulin receptors on various cells. Now, this receptor also functions as a tyrosine kinase. Kinases are enzymes that add a phosphate group, so when insulin binds to the insulin receptor, the receptor autophosphorylates, meaning that it transfers a phosphate group from an ATP molecule onto the receptor. This triggers a whole cascade of intracellular events, and the end result is that the cell mobilizes spare GLUT4 transporters, which are mostly just chilling inside the cell in small bubbles of phospholipid bilayer called vesicles. The vesicle with the transporters then moves to the membrane and fuses with it, therefore allowing more GLUT4 transporters to do their job, which speeds up glucose intake. So it's a bit like the cell's getting a love letter from the pancreas that says, "Open the door to get a sweet treat." How cool is that? Or kinda creepy. Aside from the cholesterol and proteins, cell membranes also contain various carbohydrates, which are usually bound to proteins creating glycoproteins or bound to lipids creating glycolipids. These glycoproteins and glycolipids are unique to each cell, and they help cells recognise one another, a bit like ID cards. In fact, immune cells often distinguish normal cells from invading pathogens based mainly on the glycoproteins and glycolipids on the cell surface. Now, the proteins and cholesterol within the bilayer aren't static, but they float horizontally through the phospholipids. In fact, if you look at a cell membrane from above, it looks a bit like a mosaic with parts that can move around. So that's why the cell membrane is sometimes referred to as the fluid mosaic model. Alright, so as a quick recap, the phospholipid bilayer contains cholesterol, which makes it more durable at all temperatures, as well as various proteins. Transport proteins help move molecules across the membrane, cell surface receptors receive signals from other cells, and adhesion proteins keep the cell anchored to neighbouring cells. Finally, the cell membrane contains carbohydrates in the form of glycoproteins and glycolipids, which help the cells recognise each other.
Thank you.